Hi, my name's Kate Poole and I lead the Cellular Mechanotransduction Laboratory in the Single Molecule Science Department at the University of New South Wales. And what I want to talk to you about today is cellular mechanotransduction via force sensing or mechanically activated ion channels and how this relates to how the human body might respond to changes in mechanical loading. So if you think about mechanobiology, this is basically a field that studies how our bodies adapt to and are influenced by mechanical inputs. And there are actually a surprisingly large number of physiological systems that are mechanically responsive. So our cardiovasculature, our heart needs to beat, our blood vessels will respond to changes in blood pressure, and red blood cells will actually deform depending on shear forces. Our skeletal system, our bones and our cartilage will adapt and respond to changing mechanical loads. There are mechanical elements to our digestive and respiratory systems, and even our skin and immune cells can actually sense and probe the mechanics of their surroundings. So given all of these mechanoresponsive elements to human physiology, it's actually not surprising that when you go from the persistent 1G gravitational vector into microgravity, you might see disruptions in human physiology. And this is really well described and well known. And some of the really major changes that you see are reduction in muscle mass and bone mass. There are quite significant shifts in fluid distribution in the body that could then also drive adaptive changes and changes in the sensory motor skills. So if this is all happening at the level of the organism, can individual cells, if you take them out of that organism, can they also detect changes in gravity if you go from 1G to microgravity? And what might that tell us about overall human physiology in the microgravity state? Well, there's been a number of research studies over the years that have sort of tried to describe what might happen when individual cells are exposed to microgravity. And it's clear that they can sense and respond to these changes because you do see a lot of changes in cell behavior. So the most commonly described changes that you see is if you expose cells to either real or simulated microgravity, if they're isolated cells on a surface, is that you'll start to see some adaptation in the first few minutes. And then over hours, cells will change their morphology, round up, and a subset of cells that are on that substrate will actually detach from the substrate and form multicellular aggregates, while some may stay remaining attached to the surface. So it's clear that you get these changes overall, but there's still really an open question about what are the molecular force sensors that signal changes in the gravitational state to the cells. What are the actual force sensing molecules that drive this adaptive change? This turns out to be quite a tricky question to answer. On one hand, because we're still really collecting data about what the molecular force sensors in cells are. And secondly, because the outcomes of putting different cell types into microgravity have led to a somewhat confusing picture. So one way that you might think about to approach this question of what are these molecular force sensors is to look at that set of genes where you have transcription changes between 1G and microgravity and ask whether they sit downstream of a known force sensor. But this is where we run into the first problem. So on this slide, which is admittedly very complicated, it's a condensation of a lot of different data from a lot of different papers that have looked at the change in expression of molecules that are related to the cytoskeleton, folate adhesions, cell-cell junctions, and the extracellular matrix. So all of those compartments that are involved in cellular interactions with their surroundings. And these have been done in a number of different types of cells. And in many cases, the data is being binned based on whether the cells were still adherent or in suspension. And here it's just displayed as, in the red box at the top, examples where those genes were upregulated in response to microgravity. Those genes that were downregulated, and those ones where there was no significant difference. And basically what I want you to take away from this slide here is just how complicated it is. There's no systematic changes. So you might have one molecule that is differentially regulated based on the cellular background, but also within the same cell background, say here with the prostate cancer cells, that in the suspended cells there's no change, but in the adherent cells there is. And so the take-home message from this complexity is that the responses of these cells to microgravity seem to be context dependent. So they might change based on adherent versus suspension cells, and they definitely vary depending on cell background. And so if we want to take this information and start thinking about, well, which force sensors might explain this kind of context dependency, I'd like to suggest that the mechanically activated ion channels actually really do fit this profile. And so for most of the rest of this talk, I'm going to be showing you a bunch of data that we've generated looking at the context dependency of mechanically activated ion channels that really feeds into this hypothesis. So what am I talking about when I say a mechanically activated ion channel? These are basically pore-forming proteins that sit in the plasma membrane. And what defines them as mechanically activated is that the open probability of the channel increases with some kind of increasing mechanical input. When the channel opens, you get a transmembrane flux of ions, and depending on the specific channel, it'll have a specificity for a subset of ions. What else is quite characteristic of this type of signaling is that it occurs on the millisecond timescale, so it's extremely rapid, and it's also quite localized. So how does this sort of fit with our perspective of mechanobiology and mechanical responsiveness in different physiological cells and tissues? Well, the piezo ion channels, which is the best characterized family of mammalian ion channels, and piezo 1 was really the first one that was characterized as a bona fide mechanically activated ion channel in mammalian systems, was first identified in our sensory neurons, and piezo 2 underpins our sense of touch. And there it makes a lot of sense that you would have this really rapid signaling because you need to very rapidly convert a mechanical input when something touches the skin into an electrical response that can then generate an action potential that propagates to the central nervous system. But if you look at the distribution profile of the expression profile of piezo 1 in particular, what you actually start to notice is that it's expressed in a lot of different variant cells and tissues throughout the body. And I'll highlight two main ones here. Piezo 1 is found in the bones and cartilage, and we know that these are mechanoresponsive tissues. Piezo 1 is also found in multiple elements of the cardiovascular system. So it's expressed in cardiomyocytes, cardiofibroblasts, in vascular endothelial cells, and even in the red blood cells themselves. And this suggests like quite a complex ability to transduce mechanical inputs into meaningful biological signals. This is all thinking about externally applied forces, and cells can actually really probe actively the mechanics of their surroundings. And there's evidence that the piezos can really feed into this cell-generated forces and sensing of the mechanics of the surroundings. So this is a set of molecules that are found throughout the body in a number of these different cells and tissues that are known to be affected by time spent in microgravity. So what's the picture of how these things are activated? The canonical view sort of tends to assume that our mechanically activated ion channels sit in the exposed surface of the cell, where they respond to changes in the mechanical nature of the surrounding lipids. But we think that there's also a role for these molecules to play in the cell-substrate interface, where clearly you do have other force-sensing molecules, such as the integrins and strain-gauge proteins that you find in focal adhesions. And we've also been looking at what might be happening with mechanically activated ion channels and cell-cell junctions, where again you do have other force-sensing molecules, but there is data starting to emerge with microgravity as well, that these cell-cell junctions can be disrupted in the microgravity state. So we think that this is a really interesting class of molecules to think about as potential force sensors that might modulate cellular responses to microgravity. So how do we go about actually studying these molecules in the lab? Well, if you really want to get a very precise picture of what's going on with mechanically activated ion channels, you want to directly measure their activity. There's been two main workhorse techniques that have been used over a number of years now to actually directly study that ionic flux that occurs when you mechanically stimulate a cell. The first one is based around this concept of membrane stretch. So when we want to look at ion channel activity, the gold standard technique is actually to use electrophysiology, because that way you can directly monitor the ionic flux across the membrane. And so in this picture here, that's what this element is. That's our patch clamp pipette that has a recording electrode sitting in it. And we form a very high resistance seal between that pipette and the cell membrane. We have a reference electrode that basically sits in above. And that means in this configuration, any channel that opens in this patch of membrane, you'll actually be able to measure the ionic flux that occurs across the membrane. But we need to then apply a mechanical stimulus. And here, what you do is you actually apply pressure through the patch pipette itself, and that will locally deform and stretch that patch of membrane. And so I think you can appreciate why people have really sort of made this assumption that this is how mechanically activated ion channels are in fact activated, because this was the main workhorse of showing that mechanical activation of ion channels. That's really just a sampling question of if that's the technique that you can use, then that's the insight that you gain. So a complementary technique that's also been quite widely used is, instead of just looking at this small patch of membrane that's within the patch pipette, again we're using electrophysiology, but here in this configuration you actually pop open that little bit of membrane. We call this whole cell patch clamp. And in this case we can actually measure currents that come from anywhere in the surface of the cell. So to apply the mechanical stimulus, you take a polished glass rod and indent the top of the cell. And this is actually quite a harsh stimulus, but you can actually get really good data on the mechanical activation of ion channels with this configuration. But if we want to really probe what's going on in cellular compartments beyond that exposed interface, or that exposed patch of membrane, you can see that there's not really a way to address that question with these two techniques. And so we added another technique into this toolkit, whereby we actually create elastomeric pillar arrays where we cast these cylindrical structures out of a curable elastomer, and we can culture the cells on top. And so then, again, we're in this whole cell patch clamp mode where we're monitoring any kind of channel activity throughout the surface of the cell. And to apply our mechanical stimulus, instead of indenting the top of the cell with our glass rod, we position that glass rod next to a pilus that's subjacent to the cell. And then we can apply serial deflections to that pilus. And that will then propagate a force directly to that contact point between the cells and the substrate. And that allows us to really probe what's going on at the cell-substrate interface. And just to give you a little bit of an idea of the molecules that I'm about to mention when we look at these different techniques and whether or not they might be context dependent, we'll focus mostly on piezo-1 because that is the molecule that has the most information surrounding it. There's also the related piezo-2. We're quite interested in the polymodal ion channel TRPV4, which has been implicated in a lot of different mechanosensing mechanisms. There's a stretch-activated potassium selective channel, TREC1, and then Elkin-1 is a molecule that we believe is a novel mechanically activated ion channel that we're also particularly interested in. So let's start with piezo-1 and just have a look at what happens when we apply all these three different techniques. So piezo-1 is actually activated by all of these three different types of mechanical inputs. So what we're looking at here, and I'll just walk you through this because not everyone is familiar with electrophysiological recordings. We've just got our diagram of our three techniques at the top, our pressure clamp to apply membrane stretch, indentation, and substrate deflection with our pillar arrays. And at the bottom in cyan, we have our traces from our electrophysiological recordings. And so before you've applied a mechanical stimulus, when none of these channels are open, we basically have a flat baseline, and that's telling us that we've got no current flux across the membrane. But if we apply some kind of mechanical input, those channels will start to open, and you get this macro current. And as the channels close, then that current decays. And what you can see here is that piezo-1 is activated by membrane stretch, cellular indentation, and substrate deflection. And what this suggests is that piezo-1 is actually really a broad encoder of mechanical signals that will signal to the cell rather indiscriminately different types of mechanical inputs. Now, electrophysiology is fantastic for getting really detailed information about that ionic flux, but the limitation of the technique is we get no information about the exact localization of where that flux is occurring. And so we just wanted to get a sense of whether it really was very local to the mechanical stimulus or slightly more distributed in response to a localized stimulus. And so to address this question, we used calcium imaging because part of the current flux mediated by piezo-1 is an influx of calcium ions. So in this experiment, we loaded the cells with a calcium responsive dye. And then in this dotted line here, that's the pilus that we're about to deflect. And this is sort of like an outline of our stimulator. And what you can see is in this millisecond time scale, you can start to see calcium influx at those connections right around that pillar structure. Now, by 200 milliseconds, we know from our electrophysiological recordings that actually all of those channels at the membrane are closed. So what we're seeing here in these last two frames is the downstream propagation of that calcium signal. So you can have a very localized response to the mechanical input that then is amplified downstream of that signaling. Now, there's a lot less known of how that signal is amplified downstream of mechanically activated ion channels, but we can tell that you get a very localized original response to that mechanical input. So is this going to be true for all of our different mechanically activated ion channels? Are they going to have like a similar global response to different types of mechanical inputs? Or is there more specificity than that if you look at different molecules? And when we started thinking about this question, we decided to let the biology inspire us. And we started thinking about physiological systems that experience compound mechanical loading. And a really good example of that are the chondrocytes. So we know that they're mechanically responsive in order to conduct, their physiological function. But they are also exposed to a number of different mechanical inputs. As you have joint movement, you get stretch and compression of the cells. You get fluid shear as liquid is forced into and out of the joints. And as that fluid moves, it will actually load onto the tensile elements, so those extracellular matrix molecules that make up the bulk of the tissue. And the cells will experience those forces as tensile forces directly at those contacts between the cell and the substrate. So that kind of thing that we're trying to model with our pillar array analysis. And the other thing that was really interesting about these cells when we started working on them was that they expressed two potential mechanosensors, piezo-1 and TRPV4, this polymodal ion channel. So no one had actually been able to show that TRPV4 was mechanoresponsive as a molecule itself. But when you delete it, you see all kinds of changes to mechanically driven behaviours and homeostatic processes, which really did suggest that it was a mechanoresponsive molecule. So what happens when we go back to our three different techniques and compare TRPV4 activation to piezo-1 activation? So this is just replicating our piezo-1 data. But where it gets super interesting is what happens with TRPV4. So in this high-speed pressure clamp assay where we're stretching the membrane, TRPV4 is largely agnostic to this kind of stimulus. We couldn't ever really get robust channel activation by membrane stretch. It's entirely agnostic to cell indentation, so it really never responded to indenting the cells with the glass probe. However, if we went in and had a look at what happened if we applied our stimuli directly at those cell-substrate contacts using pillar deflection, then we started to see these really beautiful mechanically activated currents. And so what this tells us is that in these chondrocytes, we actually have two mechanical sensors that are encoding different parts of the mechanical landscape. So piezo-1 is giving us this really broad overview of mechanical changes, and TRPV4 would only be signaling those tensile forces that impact the cell at connections between the cells and the surrounding substrate. Let's summarize that with a picture. Wherever there's colour here, that's suggesting that those are the mechanical inputs that that particular molecule will respond to. So piezo1 and piezo2 will respond to all these different types of input. Piezo2 is a little bit less responsive to membrane stretch, but it can still be activated by that mechanism. Whereas in contrast, TRPV4 is completely agnostic to indentation and membrane stretch and is only activated at the cell-substrate interface. Now, I can't go through the rest of the data because I don't have time for all of the other molecules that we've looked at, but just to see, TRK1, which was always known to be a stretch-activated ion channel, we could show that we could confirm in our hands that was stretch-activated, but we never saw any kind of activation by this pillar deflection. And TRK1 is also known in the literature to respond to cellular indentation. So this is yet another configuration that's the exact inverse of TRPV4. Whereas Elkin-1, which is this new molecule that we've been looking at that we think is a novel mechanically activated ion channel, Elkin-1 doesn't respond to membrane stretch, but it will encode indentations or compressions and is particularly sensitive to cell substrate deflection. So what this basically means is that mechanically activated ion channels do exhibit distinct but overlapping activation profiles, whereas an individual ion channel will respond to a different type of input. And I'd like to suggest that might mean that this class of molecules can actually engender cells with quite a tunable, exquisitely sensitive sense of touch. And this would be a way that you could see in different cellular backgrounds different outcomes in response to changes of mechanical loading or mechanical unloading like you see in microgravity, if there were just slight differences in the molecules that were being expressed. But what about outcomes? Because that's all been just thinking about the activation of the ion channel. And there's two main questions here. One is, does signaling via different ion channels lead to different outcomes? And that's quite an easy thing to conceptualize. But the other thing that I think we really need to consider is whether signaling via a specific mechanically activated ion channel is also context dependent. And what I mean by that is, if you activate an ion channel at the cell-substrate interface, is it going to do something different if you activate that ion channel in a cell-cell interface or at the exposed membrane? And that can be quite a challenging thing to tease apart. And when we started thinking about this, we were like, maybe we can just use readouts of mechanically driven cell behaviour to determine whether or not the deletion of one of these mechanically activated ion channels leads to different outcomes, depending on the context of where that channel would have been activated. And to be able to do this, we decided to use metastatic melanoma as a model. The reason that we chose these cells is because they also experience quite compound environments. So in the primary tumour site, the cells will change their own elasticity, the environment becomes stiffer, the pressure is increased as you build a tumour. But then the cells will actually dissociate from those spheroids and move through extracellular matrices and tissues of different dimensions. And so that gave us a lot of different cell behaviours where we could set it up and probe whether or not deletion of any kind of mechanically activated ion channel had more of an impact on one behaviour versus another, or whether we could see divergent outcomes if we put those cells into completely different environments. And what I'm going to show you today, we have looked at other ion channels, but for simplicity's sake, I'll just show you what happened when we deleted Elkin-1 out of one specific melanoma cell line. We wanted to create a clean knockout. So we basically used CRISPR-Cas9 to delete one of the transmembrane domains, which we know completely destroys the function. And when we look at our wild-type cells, you can see with these pillar arrays, you have these beautiful, sensitive, mechanically activated currents in our melanoma cells. And if we knocked out Elkin-1 in this particular cell line, those currents largely went away. So we have this beautiful system where we have wild-type cells, lots of mechanically activated channel activity, knockout cells, pretty much gone. But because we're using the cell behaviour as a readout, we kind of needed to confirm that we had activation of this molecule by cell-generated forces. And so again, we came back to our calcium imaging. And what you can see here is if we just let the cells probe the surface, we have like these calcium flickers that occur at the edges of the cell where it's actually pulling on the subjacent surface. And if we look at the wild-type cells, about 80% of the cells exhibit these flickers, and in the knockout cells, they were largely gone. So we know that this activity is there in response to cell-generated forces. So as we go into modelling some of these different environments that the cells experience, the first thing that we did was just put them on a 2D substrate and asked whether they had the same kind of migration properties. And in fact, the knockout cells migrate more slowly, suggesting that there's a changed interaction between cells and a two-dimensional substrate. If we complicated this situation a little bit by embedding cells in a 3D matrix, we actually lost the difference in migration speed. But if we assessed the tracks of the wild type versus the knockout, we could see that the wild type migrated further away and the knockout moved back and forth, but they're doing that at the same speed. So it seems in 2D and 3D, the deletion of alkan-1 actually has different effects on cell migration. And this hints that the context is important for the impact of this molecule on cell behaviour. But if we go from looking at cell-substrate interactions to something that's more cell-cell interaction by creating spheroids of clusters of cells and then embedding them in a matrix, we see something different again. And the data I want to show you here is what happens if we make a chimeric spheroid. So we mix equal numbers of wild-type and knockout cells, and they're just expressing different fluorophores. And what you can see is that the wild-type really forms the core of the spheroid. So this is an image of about 5,000 cells, half of which are wild type and half of which are knockout. And so the knockout cells in cyan form the outer layer of this spheroid. And we know if we just use two different colours of wild type that this is not dependent on that fluorophore, it's really driven by the wild type versus the knockout. And then if we watch them over time, what you can quite clearly see in this image, and this is after 48 hours, is that the knockout cells have dispersed away from the spheroid and invaded the matrix, whereas the wild-type cells still remain in this core. So alkan-1 regulates partitioning within and dissociation from spheroids, and it's actually quite counter to what we saw with the migration in 2D. And what that basically means is it looks from these different cell behaviours like we do have context-dependent outcomes of signalling via a single mechanically activated ion channel. And we also know that that means that mechanoelectrical transduction really does seem to be functionally relevant in multiple membrane compartments. So we still have this canonical view, but we definitely know that we can activate these channels in the cell-substrate interface. And we can also see that if you delete them from that interface, you see changes in cell behaviour. And whilst we haven't actually yet measured channel activity at the cell-cell junctions, we can see that we manipulate those cell-cell junctions when we remove one of these molecules. So the deletion of alkan-1 modulates cell behaviour in a context-dependent manner, and it acts in multiple membrane compartments. And just as a reminder, these mechanically activated ion channels exhibit distinct but overlapping activation profiles. And it's these two elements that really go back to that earlier slide that I showed you, where we have this really confusing outcome of different cells in microgravity. Because we have that sort of variation in the activation and outcomes of mechanically activated ion channels, I think this really reinforces this hypothesis that these mechanosensors might actually regulate some of these cellular responses to microgravity. And I want to show you one piece of data that we've collected that supports this contention. So we applied 4 hours of simulated microgravity using a random positioning machine, and we compared our wild type versus our alkan-1 knockout cells. And this work was done by an honors student in the lab, Daphne Wang. And what she found is if she just took this snapshot of how many cells were still adhered after that 4 hours, in the wild type, where we do have this mechanically activated channel activity, we saw a significant reduction in the number of cells left on the substrate. However, in the knockout cells, there was no significant difference between the 1G controls and the microgravity. And this is our first hint that mechanically activated ion channels may actually really be involved in cellular sensing of the gravitational state. And with that, I'd like to say thank you to the members of my team that produced some of the data that I've shown you today, Jessica Richardson, Amrita Patkunaraja, and of course Daphne Wang. I'd also like to thank Josh Chow and Julia Silvani, who are great collaborators to have, and our funding sources. Thank you very much.